Thanks, Mark, and thanks to the organizers for putting on this fantastic symposium. So good to be with friends and to make new ones today. I'd like to talk about a new technology for regenerative patch technologies. I am the chair of the DMC and also a consultant for RPT. Many of the talks today and indeed the field have talked about how we can slow the progression of age-related macular degeneration. How can we influence that? And they've looked at several monotherapies, but we're going to look today at a slightly different approach, a bioengineered implant for improving vision, not just retarding the progression in advanced dry AMD. We're going to cover a myriad of things here, from the unique design of the implant, the fact that tissue engineering and tissue replacement therapy engages multiple mechanisms of action rather than just a single molecular pathway. And we'll also talk about efficiency, engraftment, and how the design of the implant is critically important to commercialization. How does it work? As it can be done safely? How about shipping? How about manufacturing? And whether or not the data does indeed support this emerging technology. There's a robust pipeline with over 30 patents, and there's a follow-on product pipeline that we would certainly encourage all to engage in further discussion. The crux of the matter here is this unique construction which pairs a monolayer of epithelial cells and a ultra-thin Parylene membrane to replace degenerating Bruch's membrane. Clinically, we see this sharp edge area of geographic atrophy where RPE, Bruch's membrane, and downregulation of the choroid is present. And in developing this implant, we developed this monolayer of cells which have apical polarity to it. We have the implant that was manufactured under CGMP guidelines. An IND was filed in phase 1, and 2 clinical trials were fully executed with the implant procedure developed, reduced, and refined to clinical practice, and the safety monitored, which we'll discuss in the following slides. So there are 2 parts to this implant. The first, of course, is RPE cells. There's robust literature to show that pluripotential stem cells can be induced to create functional RPE cells. These RPE cells are epithelial cells, and epithelial cells crave a basement membrane. All epithelial cells crave a basement membrane because they have an apical and a basilar function, which is critical to their function, including, in this case, their critical role in the visual cycle processing. It's also important that the apical process engage with photoreceptors. They can't merely survive in the space. They have to actually have a function. We know that RPE cells is the Swiss Army knife of the retina, highly metabolic. They're nutritional, immunologic, structural, all kinds of other functions that are much more than monomodal. The ultra-thin Parylene membrane is a critical feature because we know that the Bruch's membrane is an important barrier. That's how we know angiogenesis occurs, when there are breaks in that, and it's certainly degenerated in dry AMD. So this ultra-thin membrane needs to provide a scaffold, a smooth scaffold for a monolayer of epithelial cells to attach to, but they also need to be thin enough to allow nutrients to nourish the cells, and also, of course, be permeable and allow egress of materials as well. So that's the combination. This technology here, with the graft being using a customized surgical tool to fold it, to grasp the fold, and then to insert it in the retina, is now becoming quite commonplace. The 6-step procedure with vitrectomy and targeted hydrodissection of the area of geographic atrophy, followed by a small retinotomy where the folded implant can be inserted into the eye, can be followed by a custom slow unfolding underneath the retina, followed by tamponade. So these are 3 representative examples from the series of patients. In subject 130, you can see there's a large area of geographic atrophy, larger than the size of the optic nerve, and the large RPE implant fully covers the area of geographic atrophy. That's critically important. We have all different sizes of geographic atrophy, so the ability for an implant to more fully cover all macular function is important. And you see at 1 year, there's no degradation of the implant, no shifting of the implant, and the eye is very quiet. We did see in the first several patients, some patients who developed areas of persistent bleeding and some fibrin reaction. Again, the implant did not move. It didn't appear to affect function, but it did have some persistence. And then refinement of technique, which was critically important, appeared to resolve this. And subject 403, much later, shows that even after a year, the implant remains stable and highly functional. Now, we ought to look at the safety profile because anytime you're putting a material in the eye, you want to be very concerned about whether or not there's an immunological reaction or some other trophic effect that might otherwise affect the natural history, either positively or negatively. So if you look at the OCT on the top here, you look at an area that's cut just outside where the implant, where the GA is, and then after the implant, you see in both cases there's no signs of cellular reaction. The outer retinal layers, the inner retinal layers remain unaffected. And the second question is, not only is there structurally any problems with safety, but, well, do they work? It's one thing to put cells into the space. It's quite another thing about whether or not they have adequate function. So if you look at the histologic slide, the H&E slide on the left, you'll see that the area of pigmented cells look no different. This is from actually a patient who had died from other causes 2 years afterwards. So this is 2 years after implantation. And you can see the perylene membrane and the RP pigment cells are present and they look intact without any signs of inflammation. But we also know that they produce RPE65, a visual transduction function protein. So they show that they are working. And number 3, you can see that they have a pump function. So it's not just structure and it's not just the visual transduction, but they also the ability to detergest the retina. So sodium potassium ATPase also is functional. And finally, the rhodopsin rosettes are preserved, and their phagosomes showing that RPE cells are indeed being uptaken by the RPE. So they have functional activity of this combined novel implant. So the signals of activity over years that there's some reappearance of external limiting membrane at the border, and that 7% of eyes improved, and best corrected visual acuity improved in 67%. In this graph, in the center there, you see that 27% improved by 5 letters, 40% decreased by 5 letters, but compared to the fellow eye where 80% progressed, you could say that there is some benefit to that. So in summary, we see that this can be that we have both production, surgical technique, and evidence of function, and that there's a robust patent and intellectual property profile. This is a unique opportunity for partnership for further studies, which are planned. This was just published yesterday in Translational Visual Sciences and Technology as a cover article on regenerative medicine, and there's a late-breaking session this Monday at 4 o'clock on the RPT one-year results. Thanks for your attention.